Hi, everyone. My name is Brian Rains, and I'm a principal investigator at Amicus Therapeutics and the scientific target lead for our CDD program. I'm so happy to have the opportunity today to discuss our preclinical drug development in CDD. My talk will begin with an introduction. I'll tell you about who we are and what we do. Then I'll discuss some details of our gene therapy program. Next, I'll spend a few minutes talking about our efforts to increase our understanding of basic CDKL5 biology. And finally, I'll conclude with a few notes about our protein therapy. I'd like to start by saying that Amicus Therapeutics is committed to improving the lives of patients and families affected by rare and orphan diseases. And although I know this slide looks like just a statement on a slide in a corporate presentation, as someone that's worked at Amicus now for more than 15 years, I can say that it's a deeply ingrained part of our culture and something we're proud of. Here's an overview of our drug development pipeline at Amicus, with programs from the very early preclinical stage all the way to the commercial drug Galafold for Fabry disease. As you can see, CDKL5 falls in our preclinical stage, there in purple, under the next generation research programs and CNS gene therapies. In order to tackle the significant challenges of developing a gene therapy for CDKL5 and many of those other diseases on the previous slide, we work in collaboration with the University of Pennsylvania Gene Therapy Program. And we lean on Penn's expertise in next generation capsids, animal models and behavior, and gene therapy development. We combine that with the Amicus strengths in engineering. We strive to make proteins that are essentially better than wild type by improving their expression and uptake, and using cutting-edge modeling and bioinformatics tools to design proteins that are more stable and have reduced immunogenicity. So, what is our approach for CDD? One of the main challenges for developing any drug for CNS disorder, particularly gene therapy, is the number of cells that can be corrected with essentially a limiting number of viral vectors. So we're trying to use an approach that improves the number of corrected cells without increasing the number of transduced cells. In order to do that, we're using an approach called cross-correction. We use a modified version of a viral cell uptake peptide known as TAT. TAT CDKL5, when given to CDKL5 knockout animals, has been shown to result in a phenotypic and behavioral rescue. Our gene therapy program combines secretion signals with this TAT uptake for cross-correction. And the diagram that I'm showing on the right, on the top, the figure on the top is essentially traditional gene therapy, where we are trying to transduce, infect the highest number of cells, in this case neurons, as we can. But when we combine the secretion and uptake signals with our transgene, we can turn those cells that are infected with the virus into little factories, and they start secreting the protein. And this should be taken up by nearby cells. And in that way, we can correct more cells without increasing the number of infected cells. So indeed, CDKL5 has been engineered for cross-correction at Amicus. We tested lots of different secretion signals, and we settled on a modified BIP signal sequence that promotes secretion of the transgene. We know through lots of in vitro work with our purified protein and cultured cells that TAT CDKL5 is taken up by neurons in culture. I'm showing this to you on the The images on the right, these are primary rat cortical neurons in culture for about 2 weeks, and we have treated them with our purified TAT CDKL5 for about 2 hours. And you can see, particularly in the panels on the bottom, the pink staining is CDKL5, and you can see a robust uptake throughout the cell bodies and the neurites of these neurons. Furthermore, our TAT CDKL5 is detectable in cerebrospinal fluid of treated mice. So that means that we know that when we deliver our gene therapy to CDKL5 knockout mice, we're able to see secretion of our transgene in the CSF. So the protein is secreted. We would hope that the uptake signals on the protein result in cross-correction. And so we've spent a lot of time developing ways to look for and measure this cross-correction. One of the best and prettiest, in my opinion, is this dual staining technique, where we're detecting the transgene messenger RNA, essentially where the virus is. And the image on the right, that's shown in red. And then we overlay that on the same section with CDKL5 staining for CDKL5 protein here on the right, shown in green. We then use an image analysis software that classifies each neuron. Cross-corrected neurons are identified by a positive CDKL5 protein signal, so a green signal, with no transgene messenger RNA signal. So we're looking for cells that are green and not red. I should note that the image that I'm showing here is from a section of the brain that's quite well transduced. You could argue that essentially every cell that we're looking at is likely transduced by the virus. So we're doing careful studies across the entire brain to look for less well transduced sections of the brain and see if we are able to detect more cells containing CDKL5 that have not been transduced. And that's all well and good. I can show you lots of pretty pictures, and we can learn a lot from those pictures about our drug, but ultimately they don't tell us whether the drug works. Of course, that the answer to that question, does a drug work in CDD, is more difficult than all of us would like. And to me, it's more difficult than any of the long list of diseases I've worked on in my career at Amicus. But we were, along with others, we were excited by the recent discovery of the spontaneous seizure activity in aged female mosaic mice. And we have spent a lot of time developing EEG, electroencephalograph, as a biomarker in CDD to measure whether our drug candidates work. So in the process of developing this technique, we see differences in CDKL5 knockout brain electrical activity that may precede the onset of the seizures that were described a couple of years ago. Because of these successes, we have added EEG to assess efficacy to each of our key preclinical studies in 2021. The image I'm showing here on this slide, the images are kind of our EEG setup. And you can see, despite the animals being tethered during the monitoring, they can move freely about the cage. The animals are monitored at least twice a day. And of course, the work is performed under an IACUC-approved protocol. So let's zoom in a little bit more into what we're looking for in EEG with CDKL5 knockout mice. What I'm showing is the electrical activity from essentially a 1-hour time period during the animal's dark cycle. And so the EEG on the top left, so this is from a female 3 to 4 month old control animal. This is a healthy animal. And the EEG underneath that is from a female 3 to 4 month old CDKL5 mosaic. So this is a CDKL5 het animal. And let's just to start with, let's look at just the top half of each of the EEG plots, really from 25 hertz and above. So what we're looking at here are the gamma waves. And in the control EEG on the top, the gamma waves have what we are referring to as a lot of blue sky. There's a lot of that deep blue color. And if we compare that to the CDKL5 het mouse underneath, that gamma section, the gamma band, has quite a lot of blue speckling, the lighter blue speckling, and those are gamma frequency waves. So we know, based on other studies and literature, that enhanced gamma may contribute to seizure generation. So this finding was in itself very exciting. But I would stress that we have quite a lot of other frequency, brainwave electrical frequency to monitor and perhaps find differences between those. So if we kind of decrease in frequency, we have the beta waves, which are associated with alertness and motor behavior. Underneath that, we have the alpha waves associated with relaxed wakefulness, the theta waves, learning and memory, and finally, the delta waves, which are associated with sleep. And we think that we see differences between the healthy and the CDKL5 animal in most of these frequencies. So with that in mind, this potential very powerful biomarker for testing efficacy of our drug products, we have several key studies for our CDD gene therapy. These studies are intended to prevent seizure or abnormal brainwave pathology, reverse the seizure or abnormal brainwave pathology, and we also hope to run some non-human primate safety and expression studies. Now I'll spend a few slides to talk about our efforts to increase our understanding of CDKL5 basic biology. And essentially, we're looking for proteins that interact with CDKL5 and hoping that in doing so, we can better understand the function of CDKL5 itself and also identify potential new biomarkers. So the technique that we're using to find these interacting proteins with CDKL5 is called Bioid. And I have a diagram here showing the Bioid workflow. At a basic level, we're fusing CDKL5 to an enzyme called BRA. And BRA is what's called a promiscuous biotin ligase. So if proteins interact with CDKL5, if they're in a pretty close proximity to that protein, the biotin ligase, BRA, will add the biotin molecule to those proteins. So we can take the total set of proteins, digest them, and then pull down specifically the proteins that have been biotinylated. We can then use tandem mass spec analysis to sequence those peptides, and map them onto the actual proteome of the cell. And from there, we can suggest which proteins have interacted closely with CDKL5. We've tested several different configurations of the biotin ligase, either on the N terminus or the C terminus of CDKL5, with and without the TAT cell penetrating peptide. And although I'm not ready to share the entire data set, there's still a lot of work to do. We've really only just started to scratch the surface of the protein-protein interaction analysis that we can do. There are early indications that the CDKL5 has an interesting connection to mitochondrial targets. And then one other note would be that the two centrosomal proteins, PCM1 and CEP131, are the only genes that were enriched in all the test configurations, no matter which side of CDKL5 we put the biotin ligase, or with or without the TAT cell penetrating peptide. Those two genes always showed up. Our future plans include target validation, in vivo interactome analysis. So we plan to deliver a CDKL5 BRA fusion protein to a mouse and do a very similar experiment, as well as some other proteomic tools. Now, in the last slide, I'll talk about our CDKL5 protein therapy. As I have presented in previous conferences, we spent a long time, 4 or 5 years, working on the significant challenges to express and purify milligram quantities of full-length active CDKL5. We had success when we moved to the baculoviral insect cell system and increased our expression by orders of magnitude, and simplified and focused our purification method to reduce the number of steps required to get to a greater than 90% purity. So once we did that, we spent the last year to year and a half working on developing assays, such as an in vitro kinase assay, a mass spec-based detection assay, and as I've shown already, using it to evaluate cell uptake and attempting to look at phenotypic rescue of cells in culture. But now we're at a point where we're ready to think about it as an ERT candidate more and design experiments to assess its pharmacology and look at biochemical or behavioral rescue in animal models. And currently, we're in the early stages of planning those mouse studies with purified TAT CDKL5 as ERT. And I am very much looking forward to sharing more data as it becomes available. To conclude, I'd like to thank what you can see is a long list of people at Amicus, at Penn, and at Sanford Research who we collaborated with to run the Bioid experiments. In fact, that lab invented Bioid. Without the help of all of these people, and I'm sure I'm forgetting some, we would not be where we are today with this program. We have a lot of work left ahead, but I'm extremely excited about the prospects and very much looking forward to the next year of drug development. I am happy to field any of your questions, and I'd like to thank you for your attention today. Take care.